Module 11, Experiment 11-2, Observation of a Hydra.
In this experiment, we're going to take a closer look at a very common member of phylum Cnidaria, and that's a hydra. You should have in your slide packet a prepared slide of a hydra. So we're going to take a look at this. But if you also have opportunity to go to an aquarium in your area, you might get an opportunity to take a closer look at some other cnidarians like jellyfish and sea anemones and make some illustrations of them. It's really kind of fun to watch them living specimens of these guys. So if you have an opportunity to do that, even an aquarium store, a local aquarium store might have some specimens you could look at and observe. But for this lab, we're going to take a look at the slide. And so the first thing we want to do is take a look at the hydra under 40 power, which is our smallest power. Let me get that focused and centered. All right, and once you get it centered, you want to make an illustration in your lab notebook of what you see. You can use regular pencil or color pencils if you'd like to draw what you're looking at, and there's certain things you want to label. Take a look at your figure in your book, figure 11.5, because it's going to help you identify the different parts. But some of the major parts you want to look at are the tentacles. You want to look at the mouth and label that. The basal disc, if it's visible. Take a look at a nematocyst and label the nematocyst, and also any reproductive organs that the organism might have. So do that and make sure you make a nice illustration in your lab book. And once you've done that, you want to move the slide to one of the tentacles and focus on one of those. And once you get that centered, you want to move it up to the second power, which is 100 power. Focus that. And then move to 400 power. Center your slide and focus again. And at this point, you want to take a look at the nematocyst up close. You should be able to see a lot of the nematocyst cells. You might even be able to see some little strings hanging out of the nematocysts, and those are the stingers. It's really fascinating that we have a device that helps us to see these amazing features of these tiny little creatures. So make an illustration under 400 power of the nematocyst, and then when you're done with that, you want to move your microscope power back down to 40 and recenter your slide so you can see the side of the body of your organism, because we want to focus on the side right now, the cells that are on the side of the body. Then move it up to 100 power, center it, and focus again. And then move it up to 400 power, center and focus. And then at this point, you want to make an illustration of the cells that you see on the side of the body of your organism. Again, put that in your lab notebook. Note the power that you're observing it under as well.
Another interesting cnidarian is a sea anemone. Like a hydra, sea anemones are usually in polyp form. In the last module, you read that sea anemones can live in a symbiotic relationship with clownfish. Clownfish can live among the sea anemone's tentacles without being stung. Why is that? Well, sea anemones have nematocysts on their tentacles, just like a hydra, but instead of being triggered by pressure, they're triggered by a special chemical. So if the animal comes across certain chemicals, its nematocysts will release. Now, these structures are so complex that scientists are only just beginning to understand exactly how they work, but it's believed that clownfish and a few other species of fish don't have the chemicals in their outer slime coating to cause the nematocyst to sting, yet most fish do. This is still another amazing relationship that cannot be explained via the process of macroevolution. Corals are another common group of cnidarians. They grow in polyp form and are usually very small individuals. But most corals surround themselves with a stony calcium material which is shaped like a cup. The polyp lives inside this cup. It extends its tentacles out for feeding on plankton and other organic debris, and then pulls its tentacles into the safety of its house when it's not feeding. Corals can bud to form large colonies of hard structures. As a coral dies, it can even form a new bud on top of itself, eventually creating large reefs that can grow hundreds of feet in length and many feet high. Coral reefs are an important part of warm ocean ecosystems, providing places for hundreds of other species to live. A third cnidarian I want to discuss is the jellyfish. Jellyfish commonly take the shape of a medusa form, though many also take a polyp form for part of their lives. Jellyfish usually are either male or female when they're in their medusa stage. The male releases sperm directly into the water. The eggs are in the female's gut. When a sperm fertilizes the egg, the zygote moves out of the gut and will hang on to the female's tentacles until it develops. Eventually, it forms a small swimming shape called a planula that can move around until it attaches to a hard substrate. Then it will begin to grow tentacles and a mouth, forming a polyp. While a polyp, the creature can asexually form medusas. It will create stacks and stacks of ring-like medusas until they all begin to separate off from the polyp. Because jellyfish are cnidarians, they have nematocysts on their tentacles, and these nematocysts can cause stings that can hurt humans. Now, jellyfish stings are not strong enough to outright kill you, but they can produce a very painful sting, and in some cases, if a person experiences an allergic reaction, they can cause enough problems to result in death. So it's a good idea to steer clear of them if you ever see them in the ocean.